A very warm welcome to one and all. Today's topic is on biosynthesis of triglycerides. Myself, Dr. Mausmi Shankar Adala, working as an assistant professor in the Department of Biochemistry, SJCWA, hereby brings you a brief note on what is biosynthesis of triglycerides. The contents included in today's talk is introduction, biosynthesis of TAG, monoacylglycerol, diacylglycerol, triacylglycerols in liver, triacylglycerols in adipose tissues.
Learning outcomes will be different pathways of triglycerides metabolism and their functions.
Triacylglycerols are synthesized by all the eukaryotic organisms and some prokaryotes. Although many animal cell types and organs can produce triacylglycerols, the liver, intestine, and adipose tissue are the main producers, with the latter containing the majority of body stores. Triacylglycerols are kept in the cytoplasm of all the cells, including brain cells, as lipid droplets surrounded by a monolayer of hydrophobic proteins and phospholipids, similar to oleucines in seeds or perilipins in adipose tissue. These lipid droplets are no longer thought of as a dull fat blobs, but rather as organelles with unique metabolic pathways and related enzymes. They are not exclusive to plants and animals, unlike yeast and mycobacteria. The lipid acts as a reserve of fatty acids for structural reasons or as building blocks for lipid mediators. It also stores fatty acids for energy, which can be very swiftly released as needed. Furthermore, excess bioactive and potential hazardous lipids such free fatty acids, oxylipids, and diacylglycerols, also called as cholesterol receptors, retinol receptors, and coenzyme, can be included. Esters of sequence by lipid droplets in the cells acting as a protective barrier. As a whole, triacylglycerols have no biological function. Although triacylglycerols are necessary for healthy physiology, and excessive buildup in human fat and other organs cause obesity and many health issues such as steatohepatitis, insulin resistance, and cardiomyopathy. As a result, medications that alter the metabolism and production of triacylglycerol are of great medicinal interest. Biosynthesis of triacylglycerol. There are three primary processes that are involved in the manufacture of triacylglycerol: the intestine monoacylglycerol pathway, the livers and adipose tissues, dihydroxyacetone phosphate, and glycerol 3-phosphate pathway. A fourth pathway has been identified in matured plant seeds and some animal tissues. The unique enzyme in this pathway is called diacylglycerol transferase. Kennedy pathway. More than 90% of liver triacylglycerol are created through the glycerol 3-phosphate or Kennedy pathway, which was initially identified by Professor Eugene Kennedy and others in 1950s. This process is the primary biosynthetic route to triacylglycerols. Coming to the flowchart, glycerol 3-phosphate in the presence of GPAT gives lysophosphatidic acid. GPAT stands for glycerol 3-phosphate acyl transferase. The lyophosphatidic acid in the presence of acyl glycerophosphate acyl transferase is converted into phosphatidic acid. This phosphatidic acid is in turn converted into diacylglycerol with the help of the enzyme phosphatidic acid phosphohydrolase. The diacylglycerol is finally converted into triacylglycerol with the help of the enzyme diacylglycerol acyl transferase. So this is a biosynthesis of the triacylglycerols by the Kennedy pathway included. It was long thought that the primary source of the glycerol backbone in this pathway was glycerol 3-phosphate, which is produced during the catabolism of glucose in glycolysis. However, it is known that a portion of glycerol 3-phosphate is produced de novo, that is itself, through a process called glyceroneogenesis through pyruvate, which may be the primary source in the adipose tissue. It is also possible that glycerol kinase acts as a free glycerol to a lesser extent. Much of the subsequent activity takes place in or around the endoplasmic reticulum. A glycerol 3-phosphate acyl transferase at position 1 catalyzes the esterification of the precursor glycerol 3-phosphate by a fatty acid CoA ester to form a lysophosphatidic acid. This is then acylated by an acyl glycerophosphate acyl transferase at the second position to form phosphatidic acid, a crucial intermediate in the biosynthesis of all the glycerolipids. These reactions are further explained in the lipid. These enzymes are known as to express a variety of tissues and membranes, exhibiting a wide range of isoforms and undergo distinct regulatory mechanisms. Phosphatidic acid phosphohydrolases, also known as phosphatidate phosphatases or lipid phosphatases, are a family of enzymes that then remove the phosphate group. They create 1,2-diacyl glycerols, which are the intermediates in the biosynthesis of triacyl glycerols, phospholipids, phosphatidylcholine, and phosphatidylethanolamine, as well as monogalactosylate glycerols in plants. Given that it may control the flow of lipids for storage or membrane biogenesis. This is an important branch point in the lipid biosynthesis process. Dihydroxyacetone phosphate pathway. Dihydroxyacetone phosphate in peroxisomes or the endoplasmic reticulum can be acylated with the fatty acid coenzyme A esters by an acyl transferase to form 1-acyl dihydroxyacetone phosphate, which is reduced to lysophosphatidic acid by dihydroxyacetone phosphate oxidoreductase and can subsequently enter the pathway above to triacyl glycerols. This is the second pathway for triacyl glycerol biosynthesis. Further significance comes from the precursor dihydroxyacetone phosphate, which is necessary for the biosynthesis of neutral plasmalogens. While neutral plasmalogens are not found in adipose tissue, they can play a key role in cytoplasmic droplets of many mammalian cells. Here is the map. Dihydroxyacetone phosphate is converted into 1-acyl dihydroxyacetone phosphate, further giving lysophosphatidic acid, and finally the triacyl glycerol. Monoacyl glycerol, up to 75% of the triacyl glycerols produced in the intestinal enterocytes following a meal are produced through the monoacyl glycerol route. In this, the enterocytes absorb the 2-monoacyl glycerols and free fatty acids that are liberated from dietary triacyl glycerols in the intestine. You can see below graph. There are 1,2-diacyl glycerols which are formed as an initial step in the process of acylating monoacyl glycerols by an acyl coenzyme A, monoacyl glycerol acyl transferase, and to a lesser extent by diacyl glycerol acyl transferase. However, some 2,3-diacyl glycerols, approximately about 10%, are also created. Furthermore, glycerol can be acylated and then further acylated to synthesize 1-monoacyl glycerol. In the graphical representation, you can see 2-monoacyl glycerol is converted into 1,2-diacyl glycerol and which is converted into triacyl glycerol. Triacyl glycerol metabolism in intestine, liver, and mammary gland. In Western countries, fat makes up as much as 40% of the energy consumed through the diet, with triacyl glycerols accounting for a large amount of this. Acid-stable gastric or lingual lipases initiate the process of fat digestion in the stomach. The quantity of these enzymes varies depending on the species, but in general negligible in relation to the stages that follow. But they might be necessary for effective emulsification. Bile acids, which are potent detergents that act to emulsify the hydrophobic triacyl glycerols and increase accessible surface area, are released by the gallbladder in response to the entry of triacyl glycerol breakdown products into the duodenum. This process also increases the manufacture of the hormone cholecystokinin. Triacylglycerol metabolism in adipocytes and lipid droplets. Triacylglycerol accumulations are found in adipocytes and adipose tissue. These deposits serve as the body's primary source of energy and provide insulation and cushioning for animals. Large fat deposits can be seen subcutaneously and surrounding internal organs like the liver. Each of these tissues may respond differently to metabolic restrictions. Adipose tissues also acts as a reservoir for bioactive lipids such as eicosanoids and lipid-soluble vitamins, and when necessary, provide structural components such as fatty acids, cholesterol, and retinol for membrane synthesis repair. Triacyl glycerols, which are stored when there is an excess of nutrients, are mobilized for energy production during starvation. So this is the pictorial representation where triacyl glycerol is converted into 2,3 or 1,3 diacyl glycerols and then monoacyl glycerol, and finally it is broke down into free fatty acids and glycerol. Summing up, triacylglycerols are synthesized in all eukaryotic and prokaryotic organisms. Animal cell types and organelles can produce different triacylglycerols, and these are kept in the cytoplasm of all the cells, brain cells, as lipid droplets. These lipid droplets have now came to into full existence. Initially, they are thought of being only fat blobs, but then they found out that these organelles have unique metabolic pathways and related enzymes, and then lipids acts as a reserve for fatty acids for structural reasons or building blocks for lipid mediators. It also stores fatty acids for energy, which can be swiftly released as needed. Furthermore, excess bioactive and potential hazardous lipids such as free fatty acids can be included. Thank you.